So in this objective, we assess the reproduction among the 3 different haplotypes of the potato psyllid, mainly the central, northwestern, and the western haplotype. So the main objective is to compare the reproductive performance of these 3 haplotypes on the bitter seed nightshades and the potato, because bitter seed nightshade is the overwintering host. So all these 3 haplotypes occur in the Pacific Northwest. So we included the bitter seed nightshade in our objective, so we can compare the reproductive performance of the potato psyllid haplotypes on both hosts. So the specific objectives under this main objectives were to compare the total number of eggs laid by the females in her lifetime, and the total number of the how many the eggs were hatched, and the male and the female longevity of these 3 different haplotypes on bitter seed nightshade and the potato. The second main objective of this study was to assess the interhaplotype fertility among these 3 different haplotypes. So we collected the 5th and star names from our established colonies for the 3 different haplotypes. So we collected the 5th and star names, and we put these names into the small transparent cages for adult emergence. Once the adult emerged, we collected the adults and we sexed them, and the pairs were established in those small cages. And their total lifetime fecundity, the egg fertility, and the male and the female longevity was determined from those established pairs. So here are the results of the lifetime fecundity, that how the total number of eggs per female through average total number of eggs through per female. So as you see in the first mammal, the lifetime fecundity was more than 800 eggs on the both hosts, the nightshade and the potato. I mean, whatever these 3 haplotypes, they perform equally well on the bitter seed nightshade and the potato. But when we compare the 3 haplotypes irrespective of the host plants, the northwestern haplotype and the western haplotypes are more fecund. They lay more eggs in her lifetime compared to the central haplotype. And when we looked at the egg hatch percentage, egg hatch, and we found that the egg hatch was higher, more than around 80% on the potato compared to the nightshade. On the nightshade, the egg hatch was dropped to around 61%. But when we compare these 3 haplotypes, irrespective of the host plants, all the 3 haplotypes have the similar egg hatch ratio, that's around 70 to 75%. Here is the female longevity, the total lifespan of the female. The bitter seed nightshades, the females lives longer, more than 100 days on the bitter seed nightshades compared to the potato. And when we compare the 3 haplotypes together, irrespective of the host plants, all the 3 haplotypes appear to live similar, around 100 days. There were no differences in the female longevity among the 3 haplotypes. So when we looked at the male longevity, the male longevity was also longer on the bitter seed nightshade. Is more than it's around 120 days, but on the potato it was dropped around 80 days. But when we compare these haplotypes males, irrespective of the host plants, the northwestern males appeared to be live longer compared to the western and the central haplotype. So we further looked into the fecundity and the survival together. And as you see, these dark circles are showing the fecundity, and the lines are showing the longevity of the psyllids of the three haplotypes. So as you see in this the figure, all the three haplotypes, they stop laying eggs around 90 to 100 days, but they still keep living longer on without laying eggs. And when we see the two host nightshade with the potato with the dotted lines and the nightshade with the solid lines, in the northwestern haplotypes, the psyllids live longer on both nightshades and the potato. But when it comes into the western and the central haplotype, both western and central haplotype psyllids lives longer on the bitter seed nightshades compared to the potato. So this shows that the northwestern lives on both, are more adapted to both nightshades and potato, and the western and the central haplotypes looks adapted to more adapted on the bitter seed nightshades compared to the potato. So here is the summary of this, our first objective, in which the reproduction, the fecundity differs significantly among the haplotypes, the northwestern and the western haplotypes having the highest fecundity. The egg hatch was significantly decreased when they were reared on the bitter seed nightshade. This is about 62%, and compared to the potato, which is about 81%. And northwestern haplotypes live longer on both nightshades and the potato, in contrast to the western and the central haplotypes that lives longer only on the bittersweet nightshade. The second objective of this study was to assess the inter-haplotype fertility. As you know that so far 4 different haplotypes have been identified, and we assume we don't know that either they are the same species or the different species. So the objective was that when we cross these species, if they if they interbreed, also if they interbreed or not. So for this experiment, we just collected the newly eclosed virgin males and females from the 3 different haplotypes, and their inter and intra-haplotype crosses were made, and all these newly eclosed females are unmated. So we established total 105 pairs for these inter and intra haplotype crosses. So for those established pairs, we determine the total number of eggs and how many eggs were hatched. So here are the results of our inter-haplotype fertility. So, for example, when we crossed the western female with the western males, 90% of the eggs hatched. Similarly, when we crossed the central female with the central male, 85% eggs hatched. When we crossed the central female with the northwestern female, 85% eggs hatched. Similarly, when we cross the northwestern female with the northwestern male, 85% eggs get hatched. But interestingly, when we cross these northwestern females with the western male and or central female, none of the eggs hatched. Even though the psyllids are laying a lot of, we observed around between 1,000 to 3,000 eggs per cross, but we did not find any egg hatch in this case when the northwestern females were crossed with western male and the central female. So when I did this first time, I was confused that what is going on, that why what is going on, what is happening. I did some repetition that might be I did something wrong, or there are some thrips in the cage, or accidentally if any predator get into the cage, it will destroy all the eggs. So when I further run some more experiments specifically to this cross, I find the similar results. So then I discussed these results with one of our colleague, Dr. Rodney Cooper, and during the discussion we came up with the idea that Wolbachia and the Cardinium might be the cause, because Wolbachia and Cardinium are known to cause the embryonic cytoplasmic incompatibility in the numerous insect species. So with this idea, we come on. So then we went back into the collections for the three all four haplotypes from the different locations, from Washington, Oregon, Kansas, and then the New Mexico during the different years. I mean, it's from 2005 to 2014 years, and we found that the Western haplotypes, 72% of the Western haplotypes are infected with the Wolbachia. And when we see the central haplotype, 100% of the psyllids were infected with the Wolbachia. But when it comes into the northwestern and the southwestern, none of the psyllids were infected with the Wolbachia. So we further reassures our results that we are going in the right direction. We further did some additional crossings, but these crossings are done by the Rodney Cooper lab. And similarly, they found the same trend when the northwestern females are crossed with the western and the central females. None of the eggs were hatched, and which shows that Wolbachia is the probable cause of the partial interhaplotype fertility. And as in the morning, the lady was showing that the different haplotype shifts in the different regions, might be this Wolbachia have an implication in those haplotype shifts. So here are the conclusions of this reproduction study, that fecundity differs significantly among the different haplotypes. Northwestern haplotypes and the western haplotypes have the highest fecundity. The egg hatch was significantly reduced when they were reared on the bittersweet nightshades compared to potato. The northwestern haplotypes lives longer on both nightshades and the potato, in contrast to the western and the central haplotype. And the females of the northwestern haplotypes failed to produce the viable eggs when they mated with the males of the central and the western haplotype. So this is an important implication of the Wolbachia that might be used to manage the psyllid population in the potato system. So I would like to, the finding will provided from USDA-NIFA, Washington State University, USDA-ARS, and the Northwest Potato Research Consortium.